Good day, everyone. I am Ma'am Chillet G. Credo, and I will discuss about the translation of DNA to RNA. I will also discuss about the different phases or different stages involved in polymerase chain reaction. But before that, let us tackle first the important terms in this process. So we have DNA, RNA, and proteins. So DNA, we all know that protein or DNA is known as the blueprint of life. So it is a molecule that contains biological instructions. So this biological instruction, this is the reason why every individual is unique. RNA, on the other hand, is a nucleic acid which is directly involved in protein synthesis. So its main role is to act as a messenger, conveying instructions from DNA for the protein synthesis. Okay, so the question here is, if there is DNA, why do we need RNA? So the reason behind that is that DNA is too large to go out of the cytoplasm. RNA is a single strand that is coded from the DNA. And because if this is a single strand, RNA has capability of going out the nucleus to the nuclear pores. DNA and RNA both have pentose sugar. So for RNA, the pentose sugar is ribose. And for DNA, the pentose sugar is called deoxyribose. So we have to remember that both have nitrogenous bases. However, they have one letter is different. So we can able to detect the other strand just by knowing the other strand. So RNA contains the sugar ribose phosphates and nitrogenous bases, adenine, guanine, cytosine, and uracil. So DNA, as I've said a while ago, DNA and RNA, they share the nitrogenous bases, adenine, guanine, and cytosine. However, for the DNA, thymine is usually present on the DNA, and uracil is usually present in RNA. If you know the one pair of nitrogenous base in RNA or DNA, you will be able to determine the pair of that base. So, for the complementary strand of a DNA, we have to remember the mnemonics, car in the garage and apple on the tree. Now, for the strand of RNA coded from the DNA, we have to remember the mnemonics, car in the garage and apple under the tree. So, how is that? So if the complementary nitrogen or if the nitrogenous base on one strand is cytosine, then it would be paired with guanine. Now, if the complementary base is adenine, the pair would be thymine. Okay, so for the RNA, which is coded from the DNA, if the nitrogenous base of the DNA is cytosine, then that will be paired with guanine because of the mnemonic car in the garage. So cytosine would be paired with guanine. Now, if the nitrogenous base in the DNA is adenine, that will not be paired with thymine, but rather it will be paired with uracil. Okay, so DNA resembles a twisted ladder. So remember, the sides of the ladder or the backbone contains the deoxyribose sugars and phosphates. The rungs of the ladder or the middle portion of the DNA contains the nitrogenous bases. And these nitrogenous bases, they are bonded with weak hydrogen bonds. So these weak hydrogen bonds makes the transcription process much easier. There are three important differences between the structure of DNA and the structure of RNA. So number one, the one that I had discussed a while ago, the pentose sugar found in RNA is ribose instead of deoxyribose. Number two, the nucleotide thymine found in DNA is replaced with uracil in RNA. So adenine is paired with uracil in RNA. Then number three, DNA is a double helix and RNA is a single strand. So again, thymine is paired with adenine, cytosine is paired with guanine, but then in RNA, adenine is paired with uracil, and cytosine again is paired with guanine. Okay, so let us try to answer this example. So if the sequence of one side or one single strand of DNA is CAAG, TAGGCT, what is the sequence of the complementary strand? And we are talking about the DNA. So what is the complementary strand of the DNA given this coding? So this cytosine, the complementary nitrogenous base is guanine, adenine, thymine, adenine, thymine, guanine, cytosine, thymine, adenine, adenine, thymine, guanine, cytosine, guanine, cytosine, cytosine, guanine, and thymine, adenine. The conversion of DNA to RNA is called transcription. And the production of proteins out of the RNA is called translation. So we have three types of RNA. We have messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA is the type of RNA that is produced in the transcription process. Ribosomal RNA is the RNA where messenger RNA is attached. And the transfer RNA is the type of RNA that carries specific proteins based on the codes of the messenger RNA. So what happens during the process called transcription? So in transcription, RNA polymerase breaks the hydrogen bonds between the two strands of DNA. So RNA polymerase, so what is this RNA polymerase? This RNA polymerase is an enzyme which is the precursor of the transcription process. So this RNA polymerase will stick to the double-stranded DNA and hydrogen bond will be broken. So once the hydrogen bond is broken, transcription will follow. So RNA nucleotides that are complementary to the DNA or the coding of the DNA will then be added. So what happens during transcription? So the transcription or after the transcription process. So the transcription phase of protein synthesis is over when the messenger RNA exits the nucleus through a nuclear pore and enters the cytoplasm. So during the transcription process, messenger RNA is coded from the DNA. So after this process, this messenger RNA will then go out of the nucleus through the nuclear pore. So once the messenger RNA is in the cytoplasm, then the translation process can occur. The process called translation takes place in the cellular cytoplasm. So the process is mediated by a large molecular machine called ribosomes. So after transcription, translation will proceed. The messenger RNA that carries codes based from the DNA will then be attached to the ribosomal RNA. So itong mga ribosomal RNA na to, they are like machine readers. So afterwards, transfer RNA will then be attached to the ribosomal RNA, and this transfer RNA will carry proteins based on the codes carried by the messenger RNA. Translation starts when a ribosome attaches to a messenger RNA molecule and begins to assemble amino acid based on the sequence of codons in the messenger RNA molecule. So, itong mga amino acid chain na to, or the type of amino acid chain that will be produced will be based from the codes that are based on the codes that are being found on the messenger RNA. So what are codons? So DNA and RNA use only 4 bases to create a code that determines the sequence of 20 different amino acids in the final protein. So in order to do this, nucleic acid use 3-letter words called codons to indicate each of the amino acids in the sequence. So remember, this messenger RNA is being read by 3. So 3 letters at each time, and that is called codon. Now, the codings that can be found in the codon will then determine what type of amino acid will be produced. Just like, for example, this UGG, this codon will particularly produce the type of amino acid called tryptophan. Okay, so let us see another example. So, for example, this codon 1, which is wherein the coding is GCU, this particular codon will transcribe a type of amino acid called alanine. So ACG, on the other hand, pertaining to this codon on number 2, will then produce the type of amino acid called threonine. And this codon 3, wherein the codes is GAG, these particular codings will result to the production of amino acid called glutamate. Okay, so this is the genetic code table that is being used to determine the type of amino acid that will be produced on the translation process. So for example, we have a coding, we have AUG coding. So kung AUG ang coding ng codon, then the first step is you have to look for the first letter first. So again, AUG ang coding natin. The first letter is located here, the letter A. Again, AUG, the second letter is U. Look for the second letter, so it's U. So dito siya. Then, the third letter, you will going to look at the right side of the genetic table. So letter G. So AUG, this coding will particularly produce amino acid called MET or methionine. So this is another genetic code table. So it is just the same dun sa kanina. The only difference is dito yung mga name of amino acids are present. So after discussing the translation and the transcription process, let us now discuss the polymerase chain reaction. So polymerase chain reaction, or yung tinatawag nating PCR, is a technique used to amplify small segments of DNA. And this process is sometimes called molecular photocopying. So itong PCR na to, this is a fast and inexpensive technique used to copy small segments of DNA. So significant amounts of sample of DNA are necessary for molecular and genetic analysis. So yung mga studies regarding DNA would be nearly impossible without PCR amplification. Now, in the transcription and the translation process, we had learned the natural way inside our body on how to transcribe or to copy or on how to transcribe RNA from the DNA. Now, this polymerase chain reaction, this is an artificial way of copying DNA in the laboratory, and that is being done through the polymerase chain reaction. So what is the importance of polymerase chain reaction, or what is the importance of PCR? So remember, once the DNA is amplified, or the DNA produced by the polymerase chain reaction can be used in many different laboratory procedures. For example, on the mapping techniques in Human Genome Project. Itong PCR na to is also valuable in number of laboratory and clinical techniques, including DNA fingerprinting, detection of bacteria or viruses, which is very timely, and the diagnosis of genetic disorders. PCR also has many research and practical applications. So it is routinely used in DNA cloning, medical diagnostics, and forensic analysis of DNA. So itong PCR na to, it relies on a thermostable DNA polymerase, which is called the Taq polymerase, and it requires DNA primers designed specifically for the DNA region of interest. So in the process, in the process of the polymerase chain reaction that is being done in the laboratory, this actually requires a polymerase. So in the body, we have RNA polymerase. So, di ba kanina, on the discussion of the transcription and the translation process, we had learned na ang enzyme that is precursor for the transcription process is the RNA polymerase. Now, with this artificial photocopying of the DNA, ang polymerase na ginagamit nila is yung tinatawag nating TAQ or Taq polymerase. Okay, so the DNA polymerase typically used in PCR is called Taq polymerase after the heat-tolerant bacterium from which it was isolated, which is called Thermus aquaticus. So itong Thermus aquaticus na to, they live in hot springs and hydrothermal vent. So itong DNA ng mga Thermus aquaticus or ang DNA polymerase ng Thermus aquaticus is very heat stable. So, bakit ba kailangan na heat tolerant yung polymerase na gagamitin in the PCR? The reason behind that, kasi in the process of PCR, the reaction is repeatedly cycled through a series of temperature changes. So, yung reaction would be subjected to a very high temperature. So, the reaction will also be subjected to series of temperature changes, which allow many copies of the target region to be reproduced. So, ano naman tong mga RNA primer na to? So, like other DNA polymerases, Taq polymerase can only make DNA if it's given a primer. So, a short sequence of nucleotides that provides a starting point for DNA synthesis. So, yung mga short sequence of nucleotides that provides a starting point for DNA synthesis. So, in a PCR reaction, the experimenter determines the regions of DNA that will be copied or amplified by the primers that was chosen. So, what are the steps involved in the polymerase chain reaction? So, we have denaturation, annealing, and extension. So, in the denaturation process, the reaction will be subjected to a very high temperature. So, the reason behind that is to separate or denature the DNA strands. So, this process will provide single-stranded template for the next step, which is called annealing. So, on the process or for the next step, which is called the annealing, the reaction will then be cooled down so that primers can bind to their complementary sequences on the single-stranded template DNA. For the last step, which is called extension, again, on the process or on the third step, the extension, the reaction will then will be subjected to high temperature so that the Taq polymerase can extend the primers for it to synthesize new strands of DNA. So yun po, thank you very much for listening. That concludes my discussion about the transcription and translation process as well as the polymerase chain reaction or the steps involved in the polymerase chain reaction. So thank you very much po for listening and have a good day.